In the United States, every drug must follow a regulatory pathway before it can reach patients. For professionals in regulatory affairs, understanding these pathways is essential because the choice of pathway determines the entire development strategy. Today we will explore 5 key routes: the New Drug Application or NDA, the Abbreviated New Drug Application or ANDA, the Hybrid 505(b)(2) pathway, the Biologics License Application or BLA, and the Biosimilar pathway. Each one represents a different level of innovation and a different amount of required data. Let's begin with the NDA. An NDA is used for a completely new drug, a new chemical entity that has never been approved before. This pathway requires full clinical development, starting with preclinical studies, followed by phase 1 trials for safety, phase 2 trials for dose and early efficacy, and phase 3 trials to confirm safety and effectiveness in large patient populations. In addition to clinical data, the NDA includes detailed information on chemistry, manufacturing and controls, non-clinical pharmacology and toxicology, labeling, and risk management. The objective is to demonstrate that the drug is safe, effective, and consistently manufactured. Because of the extensive data required, the NDA pathway is the longest and most expensive, but it provides market exclusivity and strong intellectual property protection. Next is the ANDA, the pathway for generic drugs. In an ANDA, the sponsor does not repeat full clinical trials because the safety and efficacy of the reference listed drug are already known. Instead, the focus is on proving pharmaceutical equivalence and bioequivalence. This means the generic must have the same active ingredient, dosage form, strength, route of administration, and comparable pharmacokinetic profile. Bioequivalence studies are usually conducted in healthy volunteers and compare parameters such as maximum concentration and area under the curve within the accepted regulatory range. Although clinical efficacy studies are not required, ANDAs undergo strict evaluation of manufacturing quality, dissolution, impurities, and stability. Now let's discuss the 505(b)(2) pathway. This is a hybrid route that sits between an NDA and an NDA. It is used when a product is not entirely new but also not a simple generic. In this pathway, the sponsor can rely partly on published literature or the FDA's previous findings for an approved drug while providing new data to support any differences. Typical examples include modified release formulations, new routes of administration, new dosage forms, or combinations of approved drugs. The advantage of 505(b)(2) is reduced development time and cost, while still allowing product differentiation and in many cases market exclusivity. Now we move from small molecules to biologics. Biologic products such as monoclonal antibodies, recombinant proteins, and vaccines are approved through the Biologics License Application or BLA. Biologics are large, complex molecules produced in living systems. Because of this complexity, the manufacturing process becomes a critical part of the regulatory review. A key principle in biologics regulation is that the process defines the product. A BLA must include detailed information on cell line development, upstream and downstream processing, purification, characterization, and immunogenicity. Even small changes in manufacturing can affect the final product, so comparability studies and process control are essential. Finally, we come to biosimilars. Biosimilars are not exact copies of biologics because biologics are too complex to replicate identically. Instead, a biosimilar must demonstrate high similarity to the reference product with no clinically meaningful differences in safety, purity, or potency. The biosimilar pathway follows a stepwise approach, beginning with extensive analytical comparability, followed by non-clinical studies if needed, clinical pharmacokinetic and pharmacodynamic studies, and at least one clinical study to confirm comparable performance. The goal is not to re-establish efficacy but to confirm similarity. Biosimilars increase competition, reduce costs, and improve patient access to biologic therapies. From a regulatory affairs perspective, selecting the correct pathway is a strategic decision. It influences development time, cost, clinical requirements, market exclusivity, and lifecycle management. An NDA may take more than a decade, while a 505(b)(2) product may take several years. An ANDA is typically faster, and a biosimilar requires extensive analytical work and a different regulatory strategy. In summary, regulatory pathways are the roadmap from molecule to market. The NDA supports innovation, the ANDA enables generics. The 505(b)(2) pathway allows hybrid development, the BLA governs biologics, and biosimilars expand access to complex therapies.